That would be wonderful. Thank you, and I'll take that. Thanks. So at the end of this class, I'm going to give you a color handout of a couple diagrams as well as a nice picture of Melvin Kelvin. If you're wondering if a guy named Melvin Kelvin came up with the Kelvin cycle, he did. I think more people need to give their children names that rhyme humor. Like I should have had like a Yolanda Orlando or a Orlando. I don't know. Anyway, you guys got a workshop. I can do better than that. Anyway, so at the end of the light-dependent reactions, what we are left with is just energy-containing molecules, ATP and NADPH, right? Does that make sense? Okay, so at the end of the light-dependent reactions, are we able to read that? I again forgot to charge my computer, so I'm desk-bound here. So the light-dependent reactions happen in the thylakoid membrane between the stroma and the lumen. The light-dependent reactions use the lumen to fill it up with H+ ions so that ATP will get produced not in the lumen but in the stroma as those H+ ions return to their equilibrium state. Does that make sense? So that's the lumen's function. So by the time we get to the light-independent reactions, we're essentially done with the lumen of the mitochondrion, of the chloroplast, sorry. Does that make sense? Yeah. The light-independent reactions are going to take place in the stroma, in the space around the thylakoids, not in the space inside them. All right, we comfortable with that? Does anyone know what we call the fluid inside the stroma? You'll occasionally see chloroplasm, all right? Which makes sense because that if the cytoplasm is the fluid inside the cell, the chloroplasm is the fluid inside the chloroplast. So specifically in the chloroplasm of the stroma is where the next bit is going to happen. And it's another cycle. But what I like is this is so different from Krebs cycle. It's very different philosophically from the Krebs cycle, but at the same time, the things that happen are going to happen here, and you're even going to see some familiar-looking molecules. Now, one nice thing is this cycle is notably a whole lot smaller. So that's nice, right? If you remember Krebs cycle, we start with oxaloacetate, right? And then we add that acetyl group, yielding us citrate or citric acid. Citrate gets metabolized into isocitrate, at which point it loses a carbon, released to CO2, it becomes alpha-ketoglutarate, right? We go through all these steps. This is comparatively a lot smaller. Does that make sense? Which makes it, A, a lot easier to use, like a lot easier to learn and memorize, but B, it's interesting stuff. So, here's what we're going to do. Is in the stroma, we have the molecule ribulose 1,5-bisphosphate. Ribulose 1,5-bisphosphate. All right, it's already present in the chloroplast. I'm going to give you a handout that has specific drawings of all these things. We're just going to track carbons, kind of like we did in our first run through cellular respiration. Does that make sense? Oh, I said it's already present, but I've written that already. I was being redundant here. This is relatively low energy sugar. It's a relatively low energy 5-carbon sugar. And what does that bisphosphate tell us about it? In addition to the 5 carbons, we have 2 phosphate groups. All right, I'm going to show those here thusly. And the fact that they're on carbons 1 and 5 means that this is, roughly symmetrical molecule. All right? And what's going to happen to it is that the RUBP, which is what we shorten ribulose 1,5-bisphosphate to, RU for ribulose, BP for biphosphate. Does that make sense? Is going to get cleaved in half. Now, how do you cleave a 5-carbon sugar in half? Well, you'd get 2 and 3, I guess. Right? Does that make sense? So you're going to get a 2-carbon sugar and a 3-carbon sugar left to itself. But we want to get it to be symmetrical. How do you do that? What could you have be also present? CO2. So this is where the carbon dioxide gets used in this process, which is we are going to have a carbon dioxide present. Does that make sense? Sorry? Oh, right. 2 oxygens, right? And we're not going to cross the carbons, right? And this is going to be a reaction that breaks something in half. Now, if it breaks something in half, does anyone remember what we call that kind of reaction? The kind of reaction that breaks a molecule apart? Yep, and we call this a hydrolysis reaction. Remember that? Where we have to put a water in to break it apart. Right? Remember, we got condensation reactions. We get a water out when we put 2 molecules together. So we're going to do ribulose bisphosphate and CO2 is going to give us 2 3-carbon sugars. All right? Yep, I'm going to give you the name in a minute. And again, I'm going to give you this diagram, but it's helpful, I think, to draw it because this one is small enough that it helps us sort of keep track. Now, this is called ribulose 1,5-bisphosphate carboxylase. We will just say RUBP carboxylase. Now, if you think about it, the reason it's so called is that both of these, and you'll see this when you get the drawing, you don't have to do it now, but each of these is a carboxylic acid. All right, each of these has good question. They're still attached, and I haven't drawn them yet. Yeah, so there we go. And we've actually seen this molecule before. These are called phosphoglyceraldehyde. And we shorten that to PGA. All right, phosphoglyceraldehyde gets shortened to PGA, and we have 2 of them because they're a 3-carbon sugar. Okay, and maybe I do want to draw the oxygen. I mean, it doesn't make sense to draw the oxygens on here because we're not tracking any of the other oxygens, so that's fine. So, where does the CO2 come from? That's right, from the air. All right, so the plant needs to bring CO2 in. The RUBP is already present. And the fact that this is a cycle means that just like how oxaloacetate was always present, because when we're done, we're going to make oxaloacetate. When this is done, we're going to remake this RUBP so that there's always ribulose bisphosphate present. Is that Charlie's hand back there? Yeah. Not that they've announced, no. Right? I didn't miss an announcement? No. Okay, so that's step one. Step one is that we're going to add a carbon dioxide, we're going to add a water, and that's going to give us glyceraldehyde, phosphoglyceraldehyde rather. Now keep in mind, this is still fairly low energy. In fact, it's probably lower energy than the ribulose bisphosphate because it's smaller, right? So where do we go now?
Attention everyone, students and staff, we are now moving out of Lock and Learn. Thank you for your cooperation. Again, thank you for your cooperation. Have a great day.
That's great. Would somebody mind hitting the back set of lights? I just don't quite have enough energy to see my own handwriting here, unfortunately. Okay, in the second stage, okay, we now have 6 carbons. Thank you. But not arranged the way we want them, right? But we still need energy to combine them into one glucose. Now, where does that energy come from? Anybody? Yeah, we've already done that. So that energy is going to come from indirectly from the sun, but where do we have it now? We're in the stroma. The light-dependent reactions have already happened. At P. Yes, at P. ATP and NADPH are going to provide that energy. Is everyone comfortable with E being energy? Yes. Okay. So let's look at that. We have our 3-phosphoglycerate, and again, for the purpose of our drawing here, we're just going to draw that this way. Is going to be reduced. If it's going to be reduced, that means it's going to be given energy, right? And if it's being reduced, then something has to be oxidized, right? So we're going to go like this, and what's going to be oxidized? In this case, ATP is going to be oxidized into ADP, and then the other phosphate group is going to get tacked on to our phosphoglyceraldehyde, and that's going to make another 2-phosphate group, and we call that 1,3-bisphosphoglycerate. All right, so this is that phosphoglyceraldehyde, and this is going to be something called 1,3-bisphosphoglycerate. 1,3, that should be a comma, that should be a dash. Bisphosphoglycerate. All right. So that reaction gives energy into this bisphosphoglycerate. Sorry? I'll give you the name after, because I definitely don't have room in this diagram to put it. And then we're going to get another paired reaction. We're going to give up energy again. And if we've already used up the ATP, what's this one going to be? NADPH is going to become what? NADP with a positive charge, right? And this is going to make a higher energy molecule called glyceraldehyde 3-phosphate. So we're actually going to give up yet a phosphate group here, but it's a higher rearrangement. So all of this is one of these things where we're just rearranging the molecules in such a way that we end up with a higher energy. All right? So, yet again, this reaction is going to give energy to that. And this one is called glyceraldehyde 3-phosphate, and this one we call G3P. Now keep in mind, this is going to happen twice, right? It's going to happen twice because from the original RUBP, how many of these did we get? 2, right? So it's going to happen twice, which means we need 2 ATPs and 2 NADPHs to drive this every time. And now, this is where it gets whatever, kind of interesting, is that 2 glyceraldehyde 3-phosphates can be combined into a single glucose molecule. Interestingly, that doesn't happen in the chloroplast. It happens just in the cytoplasm. So these G3Ps are going to leave the cytoplasm and go into the chloroplast. God, just said that backwards, didn't they? This happens outside the chloroplast in the cytoplasm. Now, you might ask, how is that a cycle? And Toby, yeah.
I was gonna say, when we're doing glycolysis, don't we get G3P?
Yeah, it shows up there. Yeah, and one of the pathways that exists, if your cell has a molecule that can take glucose and end up having it turn into G3P, all of those enzymes can work in reverse and turn G3P into glucose. Does that make sense? So it's actually some of the same mechanisms that are involved in glycolysis that will break this down. It also means that if the cell needs to use this energy right away, that G3P can enter the glycolytic pathway and just work its way down into the mitochondrion. Does that make sense? But then if it has an excess of ATP, it can enter the glycolytic pathway and work its way up and be turned into glucose. Yes, sir?
Where'd the phosphate go?
It gets released. Sorry, I should show that. It just gets released. It's free floating.
We'll use it for later.
PI, independent phosphate. And remember, there's always independent phosphates floating around, just like there's always ADP floating around, just like there's always everything else. Okay, now, how is this a cycle? Question mark. This is called the Calvin cycle. Well, the answer is that only some of the G3P leaves the mitochondrion and forms glucose. The rest of it sticks around and gets turned back in to ribulose bisphosphate. So some of it goes off to get turned into glucose. That's the net glucose that the plant gets to store. And most of it actually gets turned back into RUBP, which can go back. And so now, we're just going to write that. Only some G3P goes into the cytoplasm and forms glucose. The rest gets converted back to ribulose bisphosphate. And so now we can actually work out the specific numbers, which is for every 12 G3Ps that get produced, and this is going to be annoying, but whatever. You know I love you guys, so I'm going to do it. There's 1 of them, 2 of them, 3 of them, 4, 5, 6. Do I need to put a phosphate on every one of these? I should, eh? 7, 8, 9, 10, 11, 12. And put a phosphate on each one. That looks like trash. So every time we go through this cycle, we're going to have to make 12 G3P molecules. 2 of them can go to the cytoplasm where they will form a glucose molecule, right? What happens to the other 10? Well, how many carbons did we just get rid of? 6 of them, right? And 10 3-carbon molecules makes what? How many? 30. And this is going to get converted to RUBP. How many RUBPs can we make? Remember, RUBP had how many carbons? 5. So how many of those can we make? Good. These get converted back into 6 RUBP molecules. And they can repeat the cycle. And now we're going to draw this as a cycle, and we're going to draw it as a cycle since we know we're working in multiples of how many RUBP molecules? It takes 6 of them to do this cycle once. Does that make sense? So when we draw the cycle, we're going to draw it as 6 of them. Ready? So we start with 1, 2, 3, 4, 5 molecules that have a phosphate on both sides. This is that ribulose bisphosphate, and we actually are going to have 6 of these, right? So as we go along, what happened to the ribulose bisphosphate in the first step? It split using a CO2 molecule, and that also required water. Okay, so we're going to show that. We're going to say, okay, we need, now for every 6 ribulose bisphosphates, how many carbon dioxides do we need? 6 of them. And how many waters do we need? 6 of them, right? Bless you. Okay, what did we get after that? 1 ribulose bisphosphate became what? Bless you, I guess. Do you say bless you when somebody blows their nose? Probably not. 1 RUBP became what? 2 PGAs. So 6 RUBP is going to become how many PGAs? 12 is correct. 1, 2, 3. That's got that there. And we're going to have 12, and we call this PGA, phosphoglyceraldehyde. Now there's actually a 2-step process here. I'm not going to bother showing both steps. I mean, I guess we could, but we'll jump right to, for now at least, the 12 G3Ps. 1, 2, 3. 12 PGAs are going to need first, each one of these required what? ATP and NADPH. So we're going to need 12 NADPH, or the ATPs come first, 12 ATPs to become 12 ADPs. And then we're also going to need 12 NADPHs to become 12 NADP pluses, right? Plus H plus. And this is going to be what molecule? Yeah, glyceraldehyde 3-phosphate, G3P. And how many of those will we get? 12 of them, right? Over the course of this whole thing, how much carbon have we gained? We started with 6 times 12, so it's probably not, 6 times 5, probably not a bad idea. How many carbons do we have total here? 30 carbons. By the time we get to 12 PGAs, we have how many? 36 carbons. By the time we get to 12 G3Ps, we have 36 carbons, right? And now what can happen? Well, 2 of these we said can do what? Yeah, 2 G3Ps will go to the cytoplasm, right? And then once those 2 G3Ps have gone into the cytoplasm, what can they do? They can form a single glucose, right? 1, 2, 3, 4, 5, 6. And that's a glucose molecule. And how many carbons is a glucose? 6. Our 2 G3Ps were 6 carbons. Well, if we've taken 2 G3Ps into the cytoplasm, that means that how many of them get processed here? 10 of our G3Ps go back through the cycle, right? Oh, that's not what I want to do. And those 30 carbons that carry on are going to get converted to ribulose bisphosphate, but that conversion actually requires some energy. And so this is another energy step where every one of these is going to require an ATP to become an ADP.
Are you sure we need 12 ATP when we only have 10 of them going through the cycle? How do you get?
Yes. And the truth is that if you want to think of 2 of those as being needed to move the 2 down to the cytoplasm, that's fine. But it does require 12. The actual accounting is such that we need 12. Yeah.
For the G3P still have like a phosphate attached to them.
Yep.
So when they become glucose, do those phosphate
The phosphate would be removed along the way by the enzyme. In other words, it's going to be, you know those kinases, right? Somewhere along this process. By the way, if you look up, if you look here, stay where you are, but I'll move. If you go here, what do we notice about G3P? That's it, right? So when it goes into the cytoplasm, if you want to know what the enzymes involved are that turn it into glucose, oh my God. Great. Oh, great. If you want to know what the enzymes are that turn it into glucose, it's just glycolysis working in reverse. Does that make sense? So when the G3P enters the cytoplasm, one of them will undergo triphosphate isomerase to become a DHAP, a dihydroxyacetone phosphate. The dihydroxyacetone phosphate and the glyceraldehyde 3-phosphate will react with catalyzed by aldolase to form fructose 1,6-bisphosphate, which will then work with phosphofructokinase 1 to form fructose 6-phosphate, and so on and so forth, and work its way right backwards. Does that make sense? So the G3P is now able to just enter glycolysis working kind of backwards. That's why this goes to the cytoplasm, because the cytoplasm already has the machinery to process it. Does that make sense? This is part of the glycolytic pathway. In reverse. So it's a similar approach to a cycle where we have a bigger molecule that takes that sort of is used sort of to carry this energy along. And why did it evolve this particular way? I don't know, it just did. But now if we do some accounting here, what do we see? What we can see is that for every 6 CO2s and 6 waters and 12 NADPHs and 24 ATPs, we yield a net. Oh, sorry, what else do we need? This is all the stuff that is from outside the cycle, right? And then what's inside the cycle? We also need how many RUBPs? And 6 RUBPs, which is already present. What do we get? Well, we get 12 G3Ps, which branch off, and 2 of the G3Ps become a glucose, or potentially a fructose or whatever, but the most common mechanism is the production of glucose, but there's certainly more, right? And then 10 of those G3Ps become 6 RUBP, which you'll note is what we started with, right? That's why we can say those are already present. Okay, so can we summarize the whole process of photosynthesis in the 7 minutes that remain? I think we can. How's that feel, though? Not too bad? I think this stuff is actually easier than cellular respiration. I think there's less steps to know and less to understand. But this cycle is a little bit weird in that it kind of branches off at the end and some stuff goes one way and some stuff goes the other. That can be a little bit touchy at first. I'm going to throw a Khan Academy video up on Teams. I encourage you to watch that. I encourage you to kind of draw this out, because when it kind of clicks, you'll be like, oh, I think I get it. Yeah. Yes, I'd love to. Okay, so let's summarize photosynthesis in whole then. I think probably tomorrow I'll give you some nice color handouts because we don't have enough time for me to talk through them anyway. And then I've been told by the office I have a package here, so if it's what I'm expecting, tomorrow we're going to do a little lab where we break pigments down. Get excited. So, sorry? I mean, do you want to do finger painting with spinach leaves?
Yes.
Okay, we could probably do that. Okay. Okay, so let's take a look at this. We have the light-dependent reactions, right? And in the light-dependent reactions, what do we get? Well, we require photons, right? We require photons of energy. And for every 4 photons,
LDR is the light dependent reaction.
that's right, yep. For every 4 photons, we're going to need 2 water molecules here. All right? And we're going to release 4 electrons, which will go into photosystem 2. And I'm going to skip the cytochrome B6/F and the plastoquinone and the plastocydin and the ferredoxin We'll just focus on sort of the main steps here. But what do we produce? We produce 4 ATPs and 2 NADHs. Oh. Now, how many do we need to get a single sugar?
I thought we got NADPH.
Did I not put a P there? Shame on me. Now, how many of these does it require to make a single sugar? Well, we needed 12 NADPHs and 24 glucose. So how many runs through this are we going to need to do then? 6, right? So the other thing we could say then is that in this stage, for 6 photons, we require 12 waters, and we're going to get, not 6 photons, 6 times 4 is how many? 24, right? And we would get 24 electrons that travel through photosystem 2 into photosystem 1, and that's going to release 24 ATPs and 12 NADPHs. From there, we can go to the light-independent reactions, also known as the dark reactions sometimes. And there, we can say, oh, and I'm sorry, what gets released there? This is important. What gets released in this case? H pluses, yep, but what gets released all the way from the plant? For every 2 waters, we can release an O2, which means for every 12 waters, we can release 6 O2s. Okay, when we get to the light-independent reactions, we're going to take those 24 ATPs and those 12 NADPHs, so 24 ATPs, 12 NADHs, and now we need another outside source. We said we need 6 CO2s, right? And they react with the Calvin cycle, which we've drawn above, to produce 1 glucose. Yeah, now this is also going to require 6 waters. Now, it's actually interesting. This water here gets recovered along the way. We'll talk about this, but this gets recovered through other steps along the process. I mean, it doesn't really matter which waters you think of as being recovered, but since there's 12 here and 12 doesn't match the number in the reaction, we can think of the water we use up in the light-dependent reaction as getting recovered, but this is required new each time. And yeah, is that a lot of work to make a sugar?
Yes.
Yes, but it's a very efficient process. It works kind of like a factory. So tomorrow, like I said, as long as my supplies have come, we'll do a little lab. I'll also give you a nice handout for this. I'm also going to give you an old practice test for this one, which take with a grain of salt because I make new tests every year, and this year I am applying some differences. So I'm also going to give you a little study guide. I'm going to give you an old test. I'm also going to give you a study guide and suggest some ways in which I think your test this year is going to be a little different. But listen, especially with this unit, I'm not trying to be tricky. This stuff is complicated enough that I'm not going to throw you a ton of left field questions. It's kind of exactly what one would expect. Okay? Thank you guys. Sorry?
The test is Friday.
Tentatively, and we'll see if we need to make it Monday. It's one of these things where I don't want to rob Peter to pay Paul, but I think this is the hardest course. I think this is by far the hardest course, the unit in this course. I think by far, which by the way is the good news. I think if you make it through this unit, I think unit 1 and 2 are by far the hard part of this course, and I think it gets much easier after this. Much easier after this. Potentially Friday, maybe Monday or Tuesday. Sure.
24 ATPs just to make one group?
That's right.
Okay.
Mr. Orlando, you know the drill.